joining us once again for the Dasara lecture series, the second talk. Since its beginning in 2022, the lecture series, this annual lecture series has brought together brilliant minds to share ideas that connect research with curiosity. Through these talks, we aim to make science engaging and accessible to everyone. The Planetarium has always stood as a beacon for scientific learning and imagination, and we are delighted to carry that spirit forward this evening. We hope today's lecture inspires you and adds another spark to your curiosity about the world around us. Again, welcome you all, and I request our director, Dr. Guru Prasad, to formally welcome the gathering and also to introduce today's speaker.
On this subject, Professor Sudha Rajamani of IISER Pune has kindly agreed to come here. She has honored our request and graced the occasion, and she is going to speak on the subject from molecules to cells, the carbon connection. Okay, I'm sure it's a very, very exciting subject. I don't want to preempt anything. Exception to her. She is very much involved in outreach for a long time. Okay, let me introduce her formally. Professor Sudha Rajamani got her PhD in biochemistry from the National Institute of Immunology in New Delhi, India. She then did her postdoctoral research at the Federation of American Scientists Center for Systems Biology at Harvard University and at the Deamer Lab at the University of California at Santa Cruz, where her research interests are in astrobiology and especially in the origin of life. What an interesting topic. They were nurtured. She is currently a professor in the biology department of the Indian Institute of Science, Education and Research, that is short IISER as it is known, in Pune, India. Professor Sudha Rajamani is the principal investigator of the Chemical Origins of Life Lab, where she and her group use tools from diverse areas of biology, chemistry and geochemistry to delineate how the transition from non-life to life would have occurred on the early Earth. This is one of the most profound questions haunting humanity today. She is involved in that research. So I'm sure that all of you will make use of her talk today to get inspired from that, and who knows, a lot of budding biologists might be there. Professor Sudha Rajamani is also passionate about astrobiology education, that is more important, both formally as well as science outreach programs. In particular, she feels strongly about mentoring school and university students in their pursuit of astrobiology as a career. Having educated first in Madras and then in Delhi from the National Institute of Immunology with a PhD, she went to the United States at the University of California at Santa Cruz, and she changed her subject. Actually, she went to do research on Parkinson's disease. Then, fate had it that she changed her subject to astrobiology, which is a very, very interesting and fascinating topic. Then, having done research on that, she traveled all the way from the west coast of the United States to the east coast to Boston. She went to Harvard University, one of the most prestigious in the United States, did research there, came back to India. That is what is important. And in addition to, public lectures are not new to her. She has mentored so many students. She has, in the last 5 years, she has more than nearly 40 papers to her credit. And she has already contributed to many textbooks, proceedings, and she has guided so many PhD students, master students, bachelor students, undergrad students, okay, and even an interdisciplinary program from planets to life. Look at her team. So, kindly make use of her, even patents. She's not new to that. So, a person of her stature who is involved not only in academics, academic research, okay, at the same time she is equally passionate about communicating science, okay. So kindly make use of it, kindly listen to her and ask questions. I'm sure she would love to answer that. Once again, thank you very much, madam, for having raised the occasion again all the way from Pune for traveling here, and thank you very much, and I will ask Sharanya to announce.
Sir, I request our director to present a sapling and a memento from JNP side to the speaker. I would also like to present a sapling to our director.
Professor Sudha Rajamani is very much interested in the Indian space program too. As a token of our appreciation, we have presented a memento which is nothing but a rocket modeling. Okay? So, India's Bahubali rocket called LVM-3. Okay, please. That is the one which launched Chandrayaan 2 and 3. Thank you. Thank you very much, madam. The stage is yours.
Hello everyone, is everyone able to hear me? First of all, thank you Dr. Guru Prasad. I don't know how you made a modest CV sound so good. I wish I could have vanished actually when you were talking, but thank you very much. And as he mentioned, I feel very strongly about communicating science, and because of that, particularly astrobiology, because it's a very new area in India, and I came back to the country about 13 years ago. I might have been one of the first Planetarium astrobiologists in the country. Happy to share that the community is growing and we're having more people from a very diverse set of backgrounds join us. And as we go along this conversation, we are going to focus on not just carbon and how it plays a fundamentally central role in biology. And I'm also going to sort of give you a flavor of what that means for how we think about life's emergence on our planet. This saga is very old. It's thought to have happened about 4 billion years ago. And the hope is that by the end of this talk, you leave with a flavor for the two of them, even though this wasn't mentioned in the title that was sort of circulated. I put it out there because it has a fundamental implication for understanding how life came about on our planet, how chemistry transitioned to biology. And what do I mean by all of this should become apparent as we go further. We start out with a question. I'm an educator at heart, so I can't do talks where there's no interaction, so I really would appreciate interaction, yeah? So we start with the question first. That is, if it works. I need to go that side. Just give me a second. Yeah. So here's a picture. Yeah. What do you think this is? Very nice. This? Very nice. Who said catfish? Have you heard my talk before? Okay. I will change this picture. What are these? These are called giant redwoods. These are coastal redwoods from California. And these things just grow to a humongous size and live for thousands of years. And it is mind-boggling. This, very nice. No, T-Rex. You need to be precise. This is not any dinosaur. This is the T-Rex, yeah? What is this? Do we know who the human is? She became famous just yesterday. Machado. Maria Corina Machado, she got the Nobel Peace Prize. So everybody should know that she's the one who beat or didn't beat Trump. Okay, whatever. Now, what is the least common denominator that binds all of these living beings? Any guesses? There is a least common denominator. Who said that? I don't have chocolates to throw, but somebody said cells. Very nice. Cells. Cells. I need to do that. Yeah. Something is happening. I'm getting an echo. Maybe we can turn that mic off. मुझे echo आ रहा है. I'm having, yeah, I don't know if you people can hear it. I'm hearing the echo. So, cells is the least common denominator. And today we're going to spend time trying to understand what a cell is, what is it made up of, and how does carbon fit into all of this business. Yeah, let's jump right into it. It's the fundamental unit of all life. Those of you who have gone to school in the last probably 20 years would have seen this image because it's in most of the high school textbooks. There are two kinds of cells that you're seeing here. One is a prokaryotic cell, which is their most famous example is a bacteria. It's a unicellular entity. Here is a eukaryotic cell that we are made of. It's a much more complex entity. And both these cells fundamentally are made of what is called organelles. And the organelles in turn are made up of macromolecules, biological macromolecules. And these biological macromolecules are in turn made up of a whole bunch of atoms. And one of the foundational atom in all of these biomolecules is carbon. How can we then think about the emergence of the earliest cells on Earth? They're called primordial cells or protocells. One beautiful and readily sort of logical way of thinking about it is by looking at this continuum that you see between physics, chemistry, and biology. Biology is remotely not all-encompassing. I've just used a few examples. Here is a proteinaceous molecule. It's called cytochrome C. It's a very ancient enzyme. It plays a very important role in redox pathways, reduction oxidation pathways. Here is an RNA-based enzyme called a hammerhead ribozyme, which is an infectious viral particle. If you zoom into these molecules, what you see are monomers. Even though I've used only examples of amino acid monomers which make up protein, every single biomolecule that you see in biology is essentially a bunch of monomers coming together and getting joined by either covalent linkages to form complex long biomolecules, or in one special case, it's a self-assembly driven process to form higher order aggregates. And we'll touch upon some of these concepts as we move forward. But this slide is to drive home the point that zoom into more complex molecules, you're in the chemical space, and all of this interesting chemistry, all the elements that you see in biology now, formed way back in the interstellar medium. Anybody know what these are? Buckyballs? Any famous example?
The C60 Buckminster Fullerene.
Perfect. Correct. These complex molecules can readily form even in interstellar medium. And zoom further into these molecules, what do you have? You have a whole bunch of atoms that have come together to make these simpler molecules, which then come together to form much larger complex molecules called biomolecules. And something happens when this transition happens from chemistry to biology. Matter becomes alive, it starts behaving in different ways, and this new property that is endowed on them is called an emergent property because you cannot figure out where it came from. It's just a completely new property that is an outcome of the interaction of these more complex molecules. But fundamentally, what are the molecules made up of? Atoms, and one of the very important atoms is carbon. And throughout the talk, you're going to constantly keep seeing carbon coming up all along. But prior to us jumping and getting into some of the more detailed aspects of carbon and biomolecules, a little bit about how we think about life's evolution. Prior to life evolving on our planet, we needed to have a few things in place. One is our planet, so solar system had to form. And it's, this is thought to have happened about 4.5 billion years ago from a protoplanetary disk full of dust and gas. There's a whole set of hypotheses about how planetesimals and planets form. And during this formation, a lot of the elements got sort of concentrated in the planetary system. Roughly around the same time, Moon is thought to have formed by this very important event where a Mars-sized object crashed into Earth. The Mars-sized object is called Theia. It just mixed up, mushed up all the internal material of both these bodies and ejected a small satellite, which is the Moon, that got trapped in the gravitation of Earth and continues to orbit Earth even today. And two other very important events include two very controversial sort of processes. Controversial because, one, we do not know for sure the timeline associated with them. Two, there is also debate about whether it really happened in the manner that has been proposed for a while now. And this includes the late heavy bombardment, which is thought to have happened around 3.8 billion years ago, which bombardment happens, even to date, bombardment happens by exogenic material from the space. However, it's proposed that very early on there was a period when a lot of it happened. And where is the proof? The proof for this has been argued to be in the craters of the Moon. Earth has gotten completely modified by life, so you can't have some of these, evidences that are still visible on Moon. It's all gone on Earth. That has been argued to be one reason for why they proposed that late heavy bombardment was a prominent phase during the early history of life on Earth, which brought in a lot of interesting molecules that were produced in the interstellar medium. And this is one of the most important molecules of life, which is water. All of the water that we see on Earth today is proposed to have come from all this exogenous material that got delivered. A lot of ice crystals got delivered in these contributions. These things, all of these events would have had to be in place before the grand drama played out of the transition of non-life to life on the Earth. When did this happen? This is sort of an oldish paper, but somehow I like to still show it because it's easy to understand the timeline. This transition from chemistry to biology is thought to have happened around 3.8 to 4.2 billion years ago. Very early on in Earth's history, Earth formed, and just in the Hadean period itself, a lot of the saga played out, which set the stage for the formation of primordial life that eventually led to more complex life later. So carbon, a very important element, which is predominantly thought to be a product of extensively in organic chemistry for a few other very important reasons in addition to its abundance. Because I'll tell you something later, I won't tell you something to that thing right away because it could digress the conversation. But the things to remember is that a lot of the material that was chemistry that was happening in the interstellar medium could ride on asteroids and comets, and they got showered upon the Earth via meteoroids. When a meteoroid enters the Earth, it essentially, the stone that falls that you go and collect is a meteorite. So let me give you examples, because this is not hand-wavy business. Here are examples of two meteorites. This is the very famous one. It fell in 1969 in Victoria, in Australia. It's called the Murchison meteorite because it fell in a village called Murchison. And when they extracted the core of this meteorite and looked for what kinds of compounds this particular carbonaceous chondroid was made of. Carbonaceous chondroids are a very important class of molecules that are thought to have been very crucial to this exogenous delivery that I've been talking about. They basically found a whole bunch of molecules that you would recognize as relevant to biology. Acidic, basically amino acids, hydroxy acids, C2, C3, C4, C5 chain-linked acids. So carbon-based molecules, a lot of them could be readily extracted from the aqueous phase of these molecules. And from the organic phase of these molecules, even though it's not mentioned here, another very important class of molecules has been extracted. Any guesses? I'm saying organic phase, which means that molecule is not soluble in water, but it is soluble in an organic solvent. One very important biomolecular class does not dissolve in water. Any guesses? Somebody said that. Starts with an L. Lipids. There are four important biomolecular classes, right? Nucleic acids, proteins, lipids, and carbohydrates. So amongst these four classes, carbohydrates, nucleic acids, and proteins, for a good extent, are readily soluble in water. However, lipids, on the other hand, are a very interesting set of molecules which have a water-loving group and a water-hating group, and they do not dissolve in water. They do something else. They assemble into some higher order structures, about which we'll get to in a few minutes. But suffice to say that meteoritic delivery brought in a lot of interesting primordial molecules that set the stage for the interesting biochemistry that would play out on Earth. More recently, in 2019, in Alajuela in Costa Rica, you had this CM2 chondritic meteorite fall, and that's Murchison there. This is AZ meteorite here. You can see that very similar sets of classes, where classes of molecules were found in both these chondrites, giving us enough proof that a lot of material could have been delivered this way. One other more recent evidence comes from an asteroid return sample that was facilitated by the Japanese space agency JAXA. Any guesses what the name of the asteroid was? Sorry? Ryugu. More recently, there has been data from Bennu 2, which actually is something that the American Space Agency has been working on extensively, but I'm only giving you a flavor of one of them. So this was a Hayabusa mission that was facilitated by the Japanese Space Agency, and they basically brought back this asteroid material. And what this illustration here shows is that when scientists looked at the molecules present in this, they could find a whole bunch of molecules which actually are the primordial molecules that is used in biochemistry, including amines, which basically are what amino acids are made of, carboxylic acids. Heterocycles here are molecules that are found in a very crucial biomolecule of our cells. Do you know what they are? Can you recognize this heterocycle? I'll give you a hint. These heterocycles code for information. Nucleic acids, they are essentially nucleobases. These are very similar to the nucleobases that are found in our nucleic acid. So I hope I've convinced you that a lot of exogenous delivery has been happening that's brought in very cool molecules on the Earth, which are related to our biochemistry. In addition to that, in addition to exogenous extraterrestrial delivery, you've had a whole bunch of possibilities for terra firma synthesis, synthesis that happen on the earth, be it in gas phase, be it in water conditions, be it in interfacial spaces where minerals are found to have played a very important role. A whole set of this interesting chemistry would have readily provided material that eventually led to the formation of biomolecules. What we all are very confident about in the field is that the primordial material and the simpler monomers could have been present in the prebiotic soup. But how do you go from that prebiotic soup monomers to the more complex biomolecules has been a holy grail in the field. This is something that we have some reasonable understanding about, however, it is still not very elaborate. And every good proposal in science basically involves a very, very logical theory. There's a hypothesis that is put forth, which then is tested on. And in this particular case, the origins of life research is primarily rooted in a theory that was proposed by two scientists, Oparin and Haldane. Oparin was a Russian. Haldane is an American with a very interesting Indian connection to Bengal and Pondicherry. They independently proposed that given the right energy source, which would have been lightning in the early Earth's atmosphere, ultraviolet radiation, because early Earth was a very hostile, harsh place with very little ozone cover. So if you had the fundamental inorganic ingredients present, like methylene, dicarbon, and water being the canvas or the solvent in which all of that happened, ammonia, carbon dioxide, you could in principle have had very interesting sort of a laboratory setup in natural settings where your ocean provided the canvas for a lot of these reactions to have happened. And they said that maybe just then you would have had the energy to drive uphill reactions, which essentially led to the formation of more organic molecules, which we see in biology. Organic molecules are those molecules which have carbon in them that is bound to hydrogen and oxygen, predominantly hydrogen. Inorganic molecules are those where you do not have carbon abundantly. However, in some cases you do see carbon. Example, carbon dioxide. It's not an organic molecule. It's not bound to hydrogen, but it has carbon. But basically you go from a bunch of inorganic molecules to a bunch of organic molecules given the right conditions that could have been prevalent on early Earth. However, there's a lot of debate about what the early conditions were. Let me get to the next slide and then I'll talk to you about what variations people have thought about. They assumed that the atmosphere was highly reductive in nature. What that means is that there was an abundance of electrons to facilitate reactions. A theory is a theory is a theory till it is proved with an experiment. So the first important experiment came in the same year that something else which was ultra crucial for biology happened, 1953. Any guesses what 1953 is important for? The DNA structure was described by Watson, Crick, Rosalind Franklin and Jerry Donahue, and only two of them got the Nobel Prize. That's a different story, we can talk about it later. But Miller-Urey experiment also happened in 1953, where Stanley Miller was actually a postdoc in Harold Urey's lab, and he said, Harold Urey actually came to somewhere in, I think in Chicago, to give a talk on Oparin-Haldane hypothesis, and Miller was like, had already been reading up about it, so he was very excited to try and test it. So he welcomed him. Miller set up this very famous, here is a cartoon version of it, but here is a real version of the setup. Miller set up this primordial ocean setup in caption, where he had all the fundamental ingredients, water, ammonia, dihydrogen, and he simulated energy in two ways, high temperatures, and eventually once the vapor gets into, also provided a spark discharge via a tungsten electrode, provided even higher temperature, allowing for all the ingredients in the water vapor to react and result in interesting products. Come down this condenser where things are cooled down, now the gaseous vapors become liquid and you can very ready collect samples along the way. And you can do a time course. What happened after 1 hour, what happened after 1 week, what happened after 1 month, what happened after 1 year. And many of these experiments were conducted for a protracted period. Unfortunately, we don't do that long experiments these days, excepting in very rare cases. And what they found is that, what Miller found is that you can go from these molecules indeed to a whole bunch of carbon-hydrogen bonded base biomolecules. These are all in the monomeric space. They found initially a whole bunch of amino acids and hydroxy acids. And very early on, the techniques were much more crasser. They did tell you what there was, but you needed a lot of material to detect what there was, because they used an important technique called chromatography. How many of you here have heard about paper chromatography? All of you have done it in your schools, looks like it, because when I was in school, there was no paper chromatography. Very easy to do it in principle. So, paper chromatography is a technique which allows you to separate molecules based on their affinity for water versus their affinity for a organic solvent. So they used that technique and they figured out that there is this whole bunch of molecules. They had standards to check, because without that you cannot figure out what the molecule is. However, since then, and in this particular case, Miller assumed, again like the original operand-Haldane hypothesis, that early Earth was very reducing. But since then, a lot of geochemical evidence and modeling studies. How do you ever figure out what happened 4 billion years ago without having direct evidence? What is the oldest evidence that you feel confident about? Fossils? Yeah, because they are bony, sort of, they are evidences that are left by organisms which had bones, or plants which had other kinds of very, very strong material that didn't get degraded over time. However, molecules do not leave those kinds of signatures. So sometimes what you have to do is, you have to take what you know, you have to play around with multiple sets of parameters on the computer and try and figure out what might have been, and then simulate it in the lab and try your experiments and figure out if it's indeed happening the way the computer is predicting. So a lot of geological evidence since then has led people, to geologists in particular, to think that early Earth was probably not ultra reducing, it was mildly reducing. So in the last 7.5, 8 decades, you've had people try a variety of conditions where they started with the same set of molecules and tried it under, Miller's first student Jeff Bada and his last but one student Jim Cleese got together and used the same setup to do the experiments. And indeed they could replicate it, but now they had very high-end instruments to test what has been made. They used a very, very state-of-the-art technique called ICP mass spectrometry, where you really need molecules which are only in parts per billion amounts and you can detect them. And they've gone on to show that a whole bunch of more molecules resulted in this milieu, and many of the more complex amino acids also did show up. And there are people who said, I don't like reducing conditions, I want to do oxidizing conditions and see what happened. So they played around with all of this, and repeatedly you get the same set of molecules, albeit the yields vary depending on the conditions, but you get the same set of molecules. So what is the hypothesis? Simple precursors that were delivered via extraterrestrial delivery or via terrestrial synthesis allowed for the formation of precursor molecules, which then resulted in the formation of the building blocks of life, which then came together to form much more complex assemblies like cells. Now let's look at these molecules a little more closer in the subsequent slides. But yeah, 4 billion years is a lot of time to experiment, right? I'm not probably going to be alive beyond 80 years, and I probably might not work beyond 65. And within that itself, I feel excited about having discovered many things. So over time, a lot is getting sort of, and right now technology is developed so much that our understanding of some of these processes has vastly improved since 1953. So a lot of the pieces of this puzzle are coming into view, and it is truly exciting times to be doing research in this field called astrobiology. But let's look at why carbon is so special. Because the truth is, despite carbon being one of the top 10 abundant elements in the universe, there's more silicon on Earth than carbon. And there's silicon in the same column as carbon. So in the periodic table, so it can do a lot of things that carbon does. So why carbon? Carbon is very unique because it can bond to other carbon atoms and to other elements and form these very interesting molecules which are stable and which, it's very interesting, there's a trade-off between formation and breaking, which is called hydrolysis, and the capacity to be able to do this with many other atoms other than your own kind, which is not common with silicon. So the bonding properties of carbon atoms is fundamentally responsible for this very important consequence. So how many bonds can carbon form? 4. 4. Carbon is a tetravalent molecule. So it can actually form, it has 4 electrons in its outer shell. So 6 protons, 6 neutrons, 2 in an inner shell, 4 in the outer shell, and if you have to satisfy the octet rule, you can actually form 4 covalent linkages with another atom, be it carbon or something else. So it can form these 4 covalent bonds, and one of the most recognizable hydrocarbon that there is, and which is thought to have been very prevalent on early Earth because of the abundance of hydrogen in particular, and also because of reasonable abundance of carbon, is that of methane. And in addition to that, the ability to not just form bonds with its own self but with other elements means it just had the opportunity to explore a whole bunch of molecules that got very interesting for life's biochemistry. And what are these? Okay, let's first quickly talk about just the simpler hydrocarbons which exist in the chemical space. Because a lot of these bonds result in monomeric entities which are either straight chain hydrocarbons, branched hydrocarbons, or cyclic hydrocarbons. One of the most famous cyclic hydrocarbons is benzene. Yeah, we all know benzene very well. It's right here. And there are a few other things which I'll just point out. I'm not going to discuss them, but carbon also has this very interesting ability to form double bonds with some other atoms, both within its own class and outside of its class. It's basically a problem that results in what is called unsaturation, which makes this molecule very interesting and reactive. So you have saturated hydrocarbons, you have unsaturated hydrocarbons. These are still in the realm of reasonably simpler molecules. But carbon can do something more complex, which is forming much more complex molecules. Look at that. Anybody can tell me what this molecule is? It's a fatty acid. And it's a molecule which is a long chain hydrocarbon with a carboxylic acid at one end, yeah, and a methyl group at the other end. This is a, you're not answering. What did you say? It's an amino acid. It has an amino group, it's a carboxylic acid group, and this is the most simplest of all amino acids that's out there, which is called glycine. And it can bond with other, in this case it's already bonding with other heteroatoms like nitrogen and oxygen, but it can not just form linear molecules, but it can also cyclize to form a very important class of molecules called monosaccharides, sugars. Yeah? All of this are in our body. Fatty acids, which are present as lipids, amino acids, present as peptides and proteins, and here you have sugar molecules present as carbohydrates. And where else is sugar found? In one other biomolecular class. Tu bolega nahi. I'm very excited that you know everything, but I also want others to participate and others to get a chance, right? It's also present in, he said nucleic acids, right? Okay, you stop there. Where in the nucleic acid is it present? Sorry? What do you mean by RNA? RNA is a kind of nucleic acid. I want you to be more precise. Which part of RNA do you see a linked structure like this? DNA also you see it, correct? Which part of my question doesn't change? Phosphate is a phosphorus-based molecule. you see this ring structure in the monomer of the nucleic acid. Okay, hold on to your horses. We'll get to it in a second, yeah? Sorry about that. Let's start with the first biomolecule. Any guesses what adipocytes are? Okay, you will talk. Talk. You only Ray.
Cells which store fat.
He says adipocytes are cells which store fat. Correct? And fat is what kind of a biomolecule? It's a lipid. This is present in your liver, which is where fat is stored in the form of adipocytes. And it's stored in a very specific form, and we'll look at it in a second. So lipids comes from this Greek word called lipos, which literally means fat. Yeah? These are biomolecules which are soluble in organic solvents and not soluble in water. And they have a long hydrocarbon chain like I showed you, which he very correctly recognized as a fatty acid. And it's basically non-polar, and a whole bunch of examples exist in our life which we use on a daily basis. Fats, oils, some of the vitamins you pop or your body makes, hormones that drive processes in your very important functions in your body are all made up of lipids. They are a very important source of energy. They are a very important source of energy. There's also another biomolecule which is a very important source of energy. It is carbohydrate, which is essentially made of those sugar chains, right? But fat is very interesting because when you break a fat molecule, the amount of energy that is produced is much larger than that of a carbohydrate molecule. You need carbohydrates because you can very quickly access that energy source. But let's say you're fasting and you want to, I don't know, lose weight, or become all trim and look like, I don't know, Hrithik Roshan or Aishwarya Rai. You start fasting, and even though you don't take carbs and you stay away from carbs, your body still functions. It's not like it shuts down. Where does it get that energy from? It gets it from fats. And it is stored as triglycerides. Let's look at that structure shortly, yeah? What else does fat do? Source of energy, और क्या होता है?
They protect our organs from injuries.
Very nice. Mechanical injury. Yeah? Very good. That's mechanical insulation. What else?
They act as hormones.
They act as hormones.
They absorb shocks.
हाँ, तो mechanical हो गया. One more important thing. Mechanical is at the level of internal organs, right? They regulate body temperature. Basically, they provide an insulation on the outside. Correct. Very correct. Are we missing anything else? Because of forming more higher order important molecules like hormones, and one other very important thing, provides insulation to the body. काम कर दे बेटा. No? Yeah, there. Mechanical, no, you can't. You'll have to keep doing it. No, it's okay, I'll do it. Thank you very much. Provides mechanical insulation against vital organs. Somebody said that. One more important thing, which goes back to the answer to the very first question. Yeah, who said that? You said. It forms the fundamental component of plasma membranes. Without that boundary, there is no cell. If a cell has to be a cell, it has to have that boundary. At least in the context of our body. And very importantly, there are certain fatty acids that you cannot make, which has to be taken from outside. These essential fatty acids are also very important examples of lipids. Yeah, they form cell membranes and provide compartmentalization, which is super unique for biological processes to happen. So let's look at them a little bit closer. Yeah, what are the simple fats that we know of, lipids that we know of? Fatty acids, which you saw example, right? So I will just still show you. Here is a picture of it. Yeah, it's a long hydrocarbon chain molecule with carboxylic acid on one end and the methyl group on the other end. And key building block for lipids. What do we mean by that? The triglycerides we were talking about are the second important form of simple lipids. Here is the parent molecule called glycerol, and each such molecule can get esterified via the carboxylic acid to the 3 carbon positions on the glycerol, forming what is called, or up to 2, in one case it's the head group, up to 2 of them get esterified to form what are called phospholipids. These are double chain lipids as against fatty acids which are single hydrocarbon chain ending with a head group which can be charged. In this case, it is double chain. 2 of them are fatty acids, 1 of them is a head group. It could be of various kinds, and it results in the formation of what is called triacyl glycerol or triglycerides, which are a very important neutral molecule, which are your reserves that are stored in the adipocyte cells. Non-essential fatty acids are the ones that our body can make, but the essential ones you have to supplement it with diet. And here are the examples of the very famous omega fatty acids. Depending upon where the unsaturation first shows up, you either call them omega-3 fatty acid, this is the omega end, the methyl group end. It either shows up first at the 3rd position or at the 6th position or at the 9th position. And omega end is the methyl group end, it's not the acid end, it's not the hydroxyl COOH, it's not the carboxylic acid end, it's the other end. Yeah? So these omega-3 fatty acids, the omega-3 fatty acid which is alpha-linolenic acid, and the omega-6 and 9 fatty acid. 9 fatty acid is made by the body. These 2 are not made by the body. Where does it come from? How do you substitute it into your body?
Flaxseed and fish oil.
Very nice. Are those your 2 favorite things? There are other sources also. Walnuts. A lot of nuts are rich in these. Sorry? Fish? Yeah, he got the fish already. Pat down. Flaxseed. A lot of seeds, particularly flax seeds. A lot of your seeds that produce some of the oils that you use in your everyday cooking are also rich in some of these omega acids. There's a distribution of them depending upon the oil that you use. I didn't put that picture here, but that is readily something that you can find on the internet. Please go take a look at it. One of the recommendations for doctors are that if you want to get enough of all of these omega fatty acids in your body, you need to play around with the oil that you use for your cooking. Don't please stick only to one oil, play around. One day olive oil, one day groundnut oil, one day, sesame oil, which is my favorite, mustard oil. Play around with oil so that you make sure that you get all of the important omega fatty acids which are present in different ratios in all of these oils. Then you have the complex lipids. Thus far we saw fatty acids. These are the saturated versions, these are the unsaturated versions because there's a double bond. We saw triglycerides. Again, I'm putting it out in these structures to appreciate the fact that it's made up of what? Carbon. Predominantly carbon, right? Then you have the more complex lipids. You remember I talked about phospholipid, then we talked about the triglycerides. Triglycerides are chains, all 3 of them are fatty acids. In this particular case, the phospholipid is esterified with 2 fatty acid chains at 2 positions, and at the 3rd position you have a head group of some kind. In this case, it's a phosphatidic acid, but based on the head group, you have different varieties of phospholipids. And an extremely important molecule, steroid. This is a hormone. Yeah? Steroids are hormones. Your body makes them, but you also sometimes have to substitute it from outside if you have certain conditions. One very famous example of steroid that you know of, which is present in the plasma membrane, is cholesterol. Cholesterol is super crucial for the proper functioning of your plasma membrane. This whole notion that all cholesterol is bad is wrong. There is a balance between good and bad cholesterol, and you need cholesterol for your cells to be functioning properly. So that is your lipid drama, and I just want to leave this picture for the lipid class of molecules, right? They are not just found in your cell membranes, they are also something that you can use to make cells in the lab. These are synthetic cell-like entities which can be readily made by combining a mixture of fatty acid and the alcohol containing hydrocarbon chain called a fatty alcohol in the lab. It beautifully forms this cell-like and tubular structures which you can readily visualize under microscopy. In this case, it's fluorescently labeled with a tag. It specifically likes to go and sit with a hydrophobic moiety. this on the side here, which looks very similar in terms of the tubes and the, globular bulbous structures, is actually an extract from that Murchison meteorite that I showed you. They have been able to detect up to C12 chain fatty acids in the Murchison meteorite sample. I think if our technology gets better, maybe we can see some other longer chain lengths also, but thus far it's been C12. So that finishes lipid. Let's move to the next class. Unfortunately, it's up there. This is amino acids. It's called amino acid precisely because of this reason. It has an amino group, it has an acid group. And this particular carbon here is it's an alpha carbon, and it confers a very important property on this class of molecules. Not in this example, because in this case, the R chain is a hydrogen. Oh, actually it does. What am I saying? The R chain is hydrogen. It doesn't. If the R is hydrogen, it's a glycine. So in that one case, it doesn't do this, but in every other amino acid, it does something important. It plays the role of a chiral center, because it's a carbon atom to which 4 different sets of groups are attached. And chirality is a very, very important sort of property to life, about which I'm not going to discuss, but anybody interested, I'm happy to talk to you outside of this talk. But here are your famous 20 amino acids classified based on their R chain. If the R chain hits water, it's called nonpolar. If this is H, it's glycine, right? There's no mention of glycine because everybody thinks that glycine is the easiest because it's a H group here. Here's an alanine. Instead of H, you have a methyl group. Here is valine, where you have this 2 methyl groups, and you see how it just gets more complicated. And here you have the 3 important ringed amino acids, which have a very interesting property of absorbing light. All these ring structures, just like benzene, allow molecules to absorb light, and that has some very interesting consequences. Here are basic amino acids because they have a secondary amine group in their R chain, and here are acidic amino acids because in addition to the primary carboxylic acid, in the R chain they have a secondary carboxylic acid, and here is a whole bunch of polar amino acids whose names you can't read. Tyrosine, threonine, glutamine, they got glycine and polar. I don't know how they did that. Or they got it. Yeah, they did that. Serine, cysteine, and asparagine. These are the fundamental monomers. So if you take a string of pearls, each pearl is one monomer, and you can condense multiple monomers together to form the primary chain that finally folds into a protein that does a whole bunch of very, very important things in your body. That particular bond which holds 2 amino acids together is called a peptide bond. It is, please appreciate the fact that I already showed you one class of molecules, which is the lipid. There you have a monomer in which there are covalent linkages, but then they come together to form cell-like membranes. Other kinds of, you remember that structure that I showed you, the fluorescent round and tubular structures? Those are not a consequence of forming covalent linkages. They can readily form those structures the second you throw them in water because of a very important phenomenon called the hydrophobic effect. It's beyond the scope of this talk, so I'm not going to go through it in more detail, but the remaining 3 biomolecular classes, the monomers can be condensed together. A water molecule can get kicked out, resulting in a covalent linkage that allows them to form much longer chains. In the case of amino acids, it results in peptides and proteins, and that bond is called a peptide bond. And it results in basically the string of pearls. This is the primary sequence of any protein, which is a string of amino acids put together via this linkage called amide bond. And they can form very interesting secondary structural elements. Won't go into detail, but these elements essentially allow them to fold back on themselves and form these beautiful, gorgeous structures which actually allow them to do what they do. Their function depends upon this structure. And there are 2 important kinds of proteins that result from this higher order structure. Sorry about that. Fibrous protein and a globular protein. Can you give me an example of a fibrous protein? Very nice. तो कौन सी क्लास में है? Maybe that. numbers are all on a slightly lower end. This is collagen. Collagen forms by 3 of these. Each one is an alpha helical strand. 3 of them come together, rotate around each other, coil around each other to form this extremely sturdy monomeric unit, and several of them come together to essentially result in your collagen fibril, which makes up your skin, bones, cartilage, ligament. All of it is made of collagen. This, on the other hand, if you can fundamentally see, this is fibrillar, like a tube. This is more floppy and turned around into a globular structure. In this particular case, the shape is determined completely by the sequence of amino acids that's present, and this kind of molecule, a very important example is on the next slide, and you will not answer this question. Thank you for understanding. Results in a very important molecule which allows us to have this conversation today. Anybody know what that molecule is? Who said that? Correct. This is a hemoglobin molecule. It's made up of 2 alpha chains and 2 beta chains. These are 4 subunits in the hemoglobin. So each one is an independent polypeptide chain which can fold back on itself and form this structure. But if you zoom in, you go to the tertiary state where you can see the individual subunits, which essentially is a conglomeration of secondary structural elements called beta sheets, alpha helices, and a whole bunch of other things which I haven't mentioned. And if you zoom into them, what you finally get is the primary sequence of the protein. And all of this is made of carbon attached to nitrogen, hydrogen, oxygen, so on and so forth. This is collagen. For those interested, you can go to Nature Education and you can see the structure a little more in detail. This is one of the alpha helix. 3 of the alpha helices come together to form this triple helix, and this is sort of an end-on view of that molecule. If you make that triple helical rope and look at it, the rope is like this, and you're looking at it from this end, you can see how sturdy this molecule is. Many of these come together to form collagen microfibrils, which then form actual collagen fibrils. This super tight wrapping of all these chains is crucial for the very high tensile strength of this molecule, which is why your skin can do what it does, which is provide a very important protection barrier. We now move on to the next class of nucleic acid biomolecules, which is nucleic acids. Nucleic acids, everybody knows its importance. It's basically doing what in your body? Encoding information, and it's doing a whole bunch of other functions, but it's primarily made up of 3 things. This is what I was trying to ask. What is that? It's a sugar. Yeah? And then you have a phosphate, and then you have a heterocycle, which is called the nitrogenous base. Your information in the molecule is encoded in this nitrogenous base. There are 2 classes of these molecules, purines and pyrimidines, but they essentially, such monomers can come together, condense again, undergo a condensation reaction by loss of water molecule to form a polymer, and the polymer in this case is called a nucleic acid. The monomer is called a nucleotide. And here is an example of all 4 RNA nucleotides, because the sugar here is a ribose sugar, where this 2' hydroxyl present allows the molecule to do something very important. And all these atoms in the, in that pentagonal structure, which are not mentioned, are all carbon, but you see more carbons here. These are all carbons too, where it's not explicitly mentioned as carbon. This allows RNA to do something extremely unique, which DNA cannot do in our biology. It allows it to fold back. I'm going to tell you, this is a molecule which is called ribosomal RNA. It's present in your ribosomes, which makes proteins in your cells. I'm going to tell you that this is made up of just one single strand of nucleic acid. One giant single strand of nucleic acid, which folds back on itself, is stabilized by a whole bunch of interesting non-covalent interactions which are fundamentally crucial to biology, including hydrogen bonds, electrostatic interactions, van der Waals forces, and in this case, hydrophobic effect is not very prominent. In proteins it is, globular proteins in particular, resulting in this crazy multi-domain structure, which is one of the few subunits of your ribosome which synthesizes the very important molecule that allows us to basically live life due to all the functions that it can carry out, which is proteins. And very early on in life, it is thought that the first first organisms that came about didn't have to worry about making different biomolecules for survival because RNA can encode information like DNA and propagate it, but it can also act like a protein because it can facilitate reactions like a protein, catalyze reactions. So it's thought that primordial life probably just was RNA based, which eventually got evolved into what we see today, which is a DNA, RNA protein based life. That's RNA, but here is the very important cousin, DNA, where you're missing the 2' hydroxyl group. Can you see that? Anyways, it's in the there. That's the 2' hydroxyl on this. Because you have in a DNA double helix, one strand is in one orientation, the other strand is in the opposite orientation. And here are the fundamental nucleotides or the monomers that this DNA is made of. And here is a real crystal structure of a dodecamer. It's a self-complementary dodecamer, which Richard Dickerson and Draw basically sort of used X-ray diffraction methods to determine its structure. This is the most physiologically relevant DNA structure in our bodies. When I say physiologically relevant, it means lots of water present and in the presence of salts. Our cells have a lot of important salts that also are crucial for stabilizing these interactions. That brings me to their biological functions. We already saw that they are the fundamental ingredients that result in the formation of an informational molecule called nucleic acid. But what else can they do? What else do they do? RNA uses the information in DNA to make protein. So RNA basically regulates a very important process called transcription. The energy currency of our cell, which primarily drives all of the processes in the body, is ATP. Some cases you use GTP, CTP and UTP, but predominantly adenosine triphosphate. Many of the metabolic pathways in the cell get turned on or off based on the ratio of ATP and ADP present in the cell, in addition to amino acids also playing a very important role as sensors. You have other molecules which are called nucleotide derivatives, and they play a very important role in many of the enzymatic reactions. I'm not even going to touch upon that. But I also mentioned this. I showed you the hammerhead ribozyme very early on in one of the early pictures where I showed the important thing that connects physics to chemistry to biology. Hammerhead ribozyme is fundamentally a RNA enzyme which is found in a plant. It's a viroid. It's a plant virus which actually it uses the host machinery like your COVID virus, takes a copy of its genome into one large strand, keeps folding back on itself, forming a tertiary structure and cleaving into the smaller infectious particles. It's called self-cleavage. So it basically is an enzyme which can behave just like a protein. Final class, my favorite class. And I think everybody wants to go and get something to eat now after all this listening that you've done, energy that you've expended. Carbohydrates. I love breads. I don't know how many of you love breads in every form, Western form, Indian form, it doesn't matter. But you also have other very important Indian foods which are a good source of carbohydrates, but the good carbohydrates which you need. Super crucial, because one of the important functions of carbohydrates is to store energy, run all the metabolic processes. If you get tired, what do? You eat. Or you're hungry, you eat, because your body is giving signals saying, I need to do more functions, please eat something. And that's because this particular set of foods, many of them which you find here, which are famous in the Karnataka region, are a very important source of carbohydrates. It's a simple sugar. Very importantly, it's a polyhydroxy alcohol. Glycerol can be considered the sort of the main fundamental entity from which you can form the carbohydrates, and depending upon what I haven't talked about, which is a functional group, whether it has an aldehyde or a ketone, you have two series of carbohydrates. They're called aldoses or ketoses. This is just FYI. More importantly, the monosaccharide, which is the fundamental monomeric unit of all carbohydrate chains, is literally this molecule. I'm showing you here glucose in its linear form. 6 carbons. Yeah? What is this? Is this a ketose or an aldose group? It's an aldehyde, CHO. It's an aldose group. It has a whole bunch of carbons attached, in this case 6 carbons, attached to each other via a single bond, and several hydroxyl groups which are oriented around the carbon in a very specific manner. And depending upon the presence of whether it has an aldehyde group or a keto group, the length of the carbon backbone and the spatial arrangement of the H and the OH atoms, they can be classified in many ways, but I'm not going to talk about it. But one of the important consequences is that it results in what is called handedness. Are both of your hands just the same? Correct? They are? Mirror images. Yeah? That is called handedness. And a very important consequence of the specific arrangement results in what is called handedness, which results in a very important property of carbon called isomerism. Not even going to talk about it, yeah? Just telling you. And they come in many shapes and sizes, which dictate their functions. And we'll look at just very specific examples, and we'll be done soon in a few slides. If this moves, yeah. Fundamentally, they serve as raw materials for energy storage, and they provide structural support. I've been talking about energy storage. We all know the energy storage molecule, which is glucose. And what is glucose stored as in our body? Glycogen. These are those triglyceride molecules. They are stored as glycogen in our body, but in plants it's not glycogen. It's starch. They are very similar to each other, but they are different. They are rings in solution. As much as I've been showing you glucose as a linear molecule, in solutions they fold back on themselves and they form these ring-shaped structures. Then there is the chair and boat formation conformations, and I'm not going to talk about it, but suffice to say, linear chain doesn't exist. They exist in ring chain structures, and there's a whole bunch of carbohydrates. These are the monomers which we talked about. Examples other than glucose are fructose and galactose. Very important disaccharides which you have on a daily basis. Sucrose, your sugar. Then there is lactose and maltose. Lactose comes from where? Milk. Then you have many of these molecules come together to form much longer chains called oligosaccharides, or even more longer chains called polysaccharides. And again, all this thing repeats itself. How do you make chains of these molecules? You bring the monomers together, you knock a water molecule off, form a covalent linkage. In the case of glucose molecules, it's called glycosidic bond. Proteins way it was amino acid. In the case of nucleotides, I didn't tell you what it was, but it is a phosphodiester bond. In the case of carbohydrates, it is a glycosidic linkage. Of raffinose and stachyose, I'm not going to talk about, but I can share the structures with you. I don't even know examples. So where do you find them, or what do you do is what you're asking, no? I can't remember from the top of my head, I'm so sorry. But I will show you some examples of the very important disaccharide. What is this molecule which I said that falls back on itself and forms a ring? 2 molecules of glucose. Abhi tu bol rahi, ye kya hai? He knows everything. To batti mara ya just you remember this because you love it. Okay, tell me the chemical name of maltose. Okay, it's okay, I'm kidding, forget it. Where do you see maltose? Maltose is basically Anna. Correct? I'm pressing the wrong thing, sorry people. That's D-glucose there. It's intermediate that forms during the hydrolysis of starch. A very important intermediate, but you see it in malt and everything else that you're talking about. Again, the same 2 glucose molecules can form a different kind of a disaccharide. This is a dimer called a disaccharide. Any guesses what this is? It's cellobiose. This forms on the way of hydrolysis of starch. So just to give you a sense that in plants, starch, in animal, okay, where do you find cellulose? Very important, correct. It's found in plants. Cellulose is one of the most prominent carbon source in the biosphere. Huge amounts of carbon is stored in the form of cellulose. If you hydrolyze, and cellulose does a lot of important functions. It protects the organism in which it's present, so on and so forth. It's very structurally very strong because of these branched linkages. And I think it's a very crappy looking slide on the screen, but your inorganic carbon, which is carbon dioxide in the environment, is converted into organic carbon, which is essentially sugars stored as starch in plant via a very important phenomenon called photosynthesis. Plants use light, water, carbon dioxide from the atmosphere and convert it into food, sugar, which it stores for energy purposes and releases oxygen, a very important molecule without which we cannot survive. We take it up, eukaryotes, we put it through a whole bunch of cycles and we produce energy molecules called ATP. And some of it goes into forming glycogen, the energy molecule which is it's the starch equivalent in animals, which is much more readily usable. You walk up a hill and you're like panting and you're like energy, low on energy. All you have to do is take a quick bite of a energy bar. It's high in sugar, gives you the glucose and immediately, or your body also has it and it can be immediately used. Do we have proof of photosynthesis on the early earth? So turns out, very early on, it was the simple bacteria-like molecules that came, organisms that were formed. They are the prokaryotes. We have proof that they're about 3.85 billion years old. It says 3.7, but 3.85 billion years old. And here is a proof. So if I take the whole origin of life thing and put on a 24-hour clock, very early on, you had prokaryotes come about, which did a very important thing of oxygenating the atmosphere. Even though they arose under anaerobic conditions, they ended up populating they ended up basically providing oxygen to the atmosphere, which initially killed a lot of them, but allowed for more complex cells to arise, which you and I are made of, the eukaryotic cells. This happened about 2 billion years ago. And suffice to say that in these 8 decades since this field of prebiotic chemistry started in 1953, we have a good sense of how you go from the fundamental ingredients, a lot of which has carbons in it, to precursor molecules which are very crucial for forming the biomolecules of life. So you could have, in principle, had entities which are called protocells, which are the primordial cells that came about, a whole bunch of evidence coming from biology, coming from geology and chemistry has allowed us to understand the pieces of this puzzle, and we are able to actually concoct these entities in the lab. However, we haven't been able to completely produce life in a test tube. What other piece of information is important is how do these primordial or really simple single-celled entities actually survive in areas where you and I will die. Extremophiles, a lot of these extreme temperature, extreme acidity, extreme UV radiation, very cold regions are populated by life, but microbial life, not us. We have to wear heavy duty gear to go and survive there. But microbial life thrives. So we also take understanding from the biology of these organisms to walk our way back. On one end, you're doing a bottom-up approach to start with the fundamental molecules, make the biomolecules and assemble cells. On the other side, you're using a top-down approach of knowing what it is that biochemistry is telling you about these extreme organisms to eventually appreciate how life might have come about on the early earth under harsh conditions and how might it be present elsewhere in the universe. So this is how your earth might, this is an artistic rendition of how the earth looked 3.8 to 4.1 billion years ago. But look at what biology does to earth. It makes it the beautiful pale blue dot that we live on today. And this is actually a real picture from Yellowstone National Park. And this is a volcanic spring, geothermal spring, very high in temperature, very acidic in conditions. You put a finger, you pull it out, only the bone will be there. You put it long enough, even the bone will get digested. But these organisms thrive there. These unicellular organisms. So we are increasingly beginning to understand how life works from sort of life's fundamental principles sort of dictate how you can assemble these entities in life. We haven't yet been fully successful to make this self-sustaining chemical system in the laboratory that can encompass some of the important hallmarks of life. However, a whole bunch of us working in very diverse areas which is not mentioned here, some of them, for example, math is not mentioned here, but math underlies physics, astronomy, biology, we have something called mathematical biology. It feeds into all of these sort of sciences to allow us to ask how life came about on our planet and how is it distributed in the universe. So we in the lab try to understand exactly that. I'm not interested in the pulp, it's a cute thing, but yeah, here. How do you go from that soupy set of molecules to the primordial cells? And then just to kind of make you realize that macroorganisms were there. Some of them got wiped out by natural events, gases, and something very, very important. Once the asteroid impacted Earth, what happened? Huh?
Decrease in temperature due to the smoke.
That came later, you're right. A lot of light was cut off and temperatures came down and everything. Sorry? Exactly, who said that? Correct. It triggered a very important volcanic activity and we are sitting on it. The Deccan volcanism. Deccan volcanism was triggered by that Cretaceous ternary mass extinction event which essentially wiped out the dinosaurs, which is why apne ancestors could evolve into us. If the dinosaurs were there, we didn't stand a chance. You know how we looked when the dinosaurs were there? We looked like a mole. That's our ancestor there. It survived because it was not a dinosaur. It didn't get wiped out by that catastrophe. It could hide somewhere and then come out after things got better, evolved into humans, and all of this on this 24-hour clock, including the emergence and wiping out of the dinosaurs, all of it happened extremely close to midnight. We just came up literally on this 24-hour clock a few seconds before midnight, at the most a minute before midnight. And what have we managed to do to the planet in this very short period? Yeah, I think we're bad for this planet. But all of this understanding that we're gleaning from astrobiology is allowing us to do this, which is to send high-end technology to other planets and bodies in our solar system to understand if they hosted life in their past, or if that system can host life as we understand it, or maybe as we don't understand it. Here is your technological demonstration, but here is a very important rover called Perseverance, which is carrying a whole bunch of exciting gadgets, and very recently there was a result that came out from Mars, which has gotten us excited, but we're all cautiously optimistic, and we really are trying to understand, and unless we get back the samples, we will never know if it is a consequence of biology, whatever we're seeing there. But what are the other habitable worlds in our neighborhood, in our own solar system? You have Europa, you have Enceladus, this is Earth of course, you have Mars. These two are moons. Europa is a moon of Enceladus. And what is very interesting is, even though you don't have water on these worlds, you have a hydrocarbon lake. So is water the only solvent in which life could have played out? On Earth, the biochemistry that played out was completely water-based. Is it an absolute essential? Debatable. We have to do a whole bunch of experiments to figure that out, but we haven't been able to find one single alternative to water to date, which can do all of this amazing biochemistry that happens in our cells. And the hope is, before I retire, maybe, just maybe, we'll have concocted life using a completely bottom-up approach into that. Nothing is possible without a whole bunch of exciting individuals who are excited about the work that they do. And this is the various people who have joined me on this expedition over the last 13 years. Thank you for your attention. Happy to take questions. Sorry, I went 15 minutes over my time. But I talked about two things, carbon and life.
If you have questions, please raise your hands. Ask it over the mic.
First question is always difficult to come. Then how you affect you. Please.
Simple questions. Does all of these things you explained is also related to Big Bang that we know, or right?
You're saying if all of this is related to Big Bang, correct? Yeah. No Big Bang, no universe. No universe, no hydrogen, helium. No hydrogen, helium, no subsequent higher order elements like carbon, oxygen, nitrogen, lithium, and all of those things. Big Bang was fundamental to all of this. That happened way back, about over 13 billion years ago. A lot of initial Big Bang synthesis led to the formation of all of the hydrogen and most of the helium that we see today in the cosmos. And further sort of nucleosynthesis that happened in the stars, in the core of the stars, led to the formation of other elements. So, wo nahi hai to kuch aur nahi hai, as far as I'm concerned. That's the truth actually. There was a child there, huh? We need to give him the mic at some point. No, you can go. The child can be next.
Ma'am, what is chromography?
Okay, sorry, he beat you. Chromography. You mean chromatography? Chromatography literally means to be able to separate colors. Literally, right? But fundamentally, chromatography is a bunch of techniques where you can play around with a whole set of chemicals that allows you to separate molecules on some fundamental properties that they are endowed with, based on their shape, based on their charge, based on what they can interact with. You can play around with that and separate molecules. So, sorry, go ahead.
Separation of colors and molecules.
What are colors made of? Molecules, you know. So, sometimes when you use the technique, even though originally chromatography was predominantly possible only with things that were visible, nowadays chromatography is possible where you don't, for example, I used to work on blood. Yeah? I'm not a vampire. I used to work on blood because I worked on sickle cell anemia. And one of the ways I isolated the 2 alpha chains and the 2 beta chains was to use a special chromatographic technique that allowed me to separate the 2 alpha chains and the 2 beta chains. Because blood has its own color, I could very readily visualize it on the column that I was using. However, if I work with, say, serum, which is only proteins, you don't have any visual color because they're not absorbing light in the visible wavelength. You can't use naked eyes to look at this, but you have detectors which can detect certain other properties of the molecules. For example, your proteins are made up of those whole bunch of cyclic amino acids which you saw, tryptophan, tyrosine, phenylalanine. These 3 molecules absorb light in a certain wavelength and emit it at a certain other wavelength. So you can use detectors which allow us to look at it in a very, very resolved manner, and then you can visualize ki, acha maine is time pe protein dala and its X is coming out at this time, Y is coming out at this time, so on and so forth. All of this is very computerized. Now, if we want to do, which grade are you in? 9. Okay, when you go to high school, you're going to be doing separation of amino acids. Have you done it yet? You haven't. So, amino acids, I will promise, do not have a color. You cannot visualize them with naked eye. However, when you get it to react with a molecule called ninhydrin, there is a certain color that results which can be visualized. So when you apply these amino acid solutions onto a paper and run them using a bunch of solvents where you have a mix of water and water which is aqueous phase and organic solvents which provide the organic phase, you can separate the amino acids based on all their properties that I showed you. Some hate water, some have charges, so some can very readily form hydrogen bonds with water and will just... Paper chromatography works because paper is made of cellulose. Cellulose is a carbohydrate. It has a ton of what groups? Hydroxyl groups. You can very readily hydrogen bond with and I put the solvents at the bottom and let the solvents rise due to capillary action, those amino acids, between the presence of water and the paper, it just sticks tightly to the paper and stays there. But the ones which hit water will keep running up the paper. So based on these properties, I can use paper chromatography to separate the amino acids on paper. However, when this test is done, when this experiment is done, when I take it out, I see nothing. But I can use something called an anhydrin spray, which I can spray on the paper, and amino acids का एक particular group binds to anhydrin to result in a complex, which you can very readily visualize as a blue color, excepting for proline. Every other amino acid gives a blue color. So you just spray it, you put it in an oven, and in 5 minutes, 7 minutes, it develops this color. And with control standards, which you already know from company, you buy them, glycine, alanine, valine, you run them also on the paper, and then you figure out which spot is what amino acid. So chromatography allows you to separate molecules based on several of their properties.
Can we try this at home?
You can. I can share with you an experiment. There are some of these things that are available. IISER Pune has a science activity center, and it allows you to do chromatography experiments at home, having colors extracted from natural products very readily. I'm happy to share that link with the Planetarium folks, and I can ask them to pass it on to you. And there are a whole bunch of other fun experiments you can do at home with minimal things.
Thank you, ma'am.
Madam, it was very nice that I was able to get a lot of things from you. Normally, we have been told that the main polluter is carbon. There is so much of thing, like pollution means, removal of carbon. From what I could see, that carbon is very supportive to life. But the way the things are shown nowadays, that removal of life is sustained only by hydrogen and oxygen. But you definitely threw more light on the existence of carbon as an element which supports life.
Correct.
I just want to know more about it, and also the existence of graphite, a soft one, in the element in a diamond. So two extremes in one element. That is also very surprising.
Yeah, they are called allotropes of carbon. They are made up of the same element and differ fundamentally only in their structure because of the way in which they form. You can please sit down. Diamond, for example, forms at extremely high temperatures and pressures. So if you take your graphite, yeah, and you actually subject it to those temperatures and pressures, you will eventually get diamond. However, you and I cannot make diamond from that. It's not very easy. But naturally, if you see, the graphite is a allotrope of carbon which results from a process which happens at a much lower temperature and much lower pressure, which changes its nature. It is way more brittle. Take our pencil ka tip, no? I take a sutti, a hammer, and I hit it will become powder. Diamond is not easy to break because of the lattice structure that results in the diamond molecule, which allows it to have a very high tensile strength, which is why you use it for abrasive technologies or for drill bits and stuff like that. So allotropes, and this fullerene molecule is also an allotrope of carbon. However, the way in which the atoms are bonded in the three-dimensional space is very different. These fullerenes are formed basically as spheroids or as ellipsoids, and the way in which the fundamental carbon framework is present in these molecules is very different, which changes the eventual properties of the molecule. So I hope that will answer your question. But yes, to go back to what you said, fossil fuels, the rate at which we are depleting them and the rate at which we are putting out carbon dioxide due to anthropomorphic activities is not sustainable. I'm trying to find a slide which I had, if I can get to it in a second. It actually shows the amount of carbon present in natural sources, how much of it comes from various processes. Give me 1 second. I can't see with my chashma on. I didn't put it because I felt like already I'm telling too much. And here's the carbon cycle. Ah, crap, I took it out. I'm sorry. I can again share this with, share it with these folks and they can pass it on to you. It's a NASA compiled illustration from actual data. What you'll see is the number of gigatons that is present in the form of several sort of reserves, be it in the soil, be it in, in the form of hydrocarbon reserves, in water. The rate at which they're getting depleted is so quick because of our greed and our need and everything else. There are lots of other runaway processes that are getting sort of triggered, which is not good for the planet. We could see a very early ice age at the way we are sort of emitting greenhouse gases, and a whole bunch of our activities is also leading to the release of methane into the atmosphere, which is also causing another set of problems. That's also a carbon-based molecule, but it's a hydrocarbon. So carbon is important, but how we deplete the natural reserves is something that we should be extremely careful about. So yeah, you're right. It's not just hydrogen and oxygen. Carbon is also central to life. But how the cycle is happening on the planet, where some of these very important things need to be plugged or slowed down, that is what we're talking about. Because even that small change in temperature, which we think like 2 degrees mein kya farak padega? Bahut farak padega. It's going to be devastating if we don't stop it.
1 minute. No, you can ask questions. That is not the thing. The gentleman had not come yesterday. About diamond, about graphene, everything. In fact, it is available on YouTube. You can, this one, to your question. And first part of your question, to that, next Saturday, we have a lecture on carbon and climate change.
Please, yes, absolutely. You should come. I had a very important slide. I don't know where I put
We
it.
have 2 more lectures. One is carbon and climate change related things by Professor Sebastian Peter of IISER, and another on carbon and organic chemistry. Madam Professor dealt with a lot in during her lecture, but anyway, specific thing, carbon and its compounds, organic chemistry. Next Saturday, kindly go to this next Saturday. Carbon and climate, carbon and organic chemistry. Okay, you can ask.
Here, I'm unable to understand. Let me keep my scientific way of understanding. The diamond and the graphene. My child asked me a question. This is also carbon, this is also carbon. So, if you put up a thing that I want so much pressure, I can put on a carbon. So, what characteristic that changes the thing 10 carbon atoms? I want that much of diamond only. What that changes the carbon to become a diamond? Is it compresses?
It is
What
both.
temperature?
It is both the temperature and the pressure.
Is reduced? Atomic size is
The lattice structure that is formed is very different between graphite and diamond. In diamond, it's a cubic lattice that results in the formation of this particular molecule, complex molecule. Graphite, on the other hand, is a hexagonal lattice. And the way they respond to other stimuli and other processes, physical processes, is very, very different. Just the presence of this repetitive cubic lattice in diamond makes it the hardest material known to human. But graphite is not the same. The hexagonal lattice changes the way graphite behaves when you subject it to very high temperatures and pressures.
Is it like a diamond capacity, hardness? Graphene also sometimes I heard it's more.
It is, but it's not like that. Not comparable. Very, very, very different. Literally, it's your pencil tips. I can take a hammer and powder it. I can't take a real diamond which has not got any impurity in its lattice and readily powder it. Not possible. And then there's a girl there. And then there's a kid here.
First 2 questions.
Ma'am, the session was very, very interesting. My question
You know
is,
everything, right? But yeah, go ahead.
my question is it possible for silicon-based life to exist other than the Earth in different conditions, like in different solvents?
Possible, we do not know. What we do know, at least from silicon's chemistry that we see on Earth, is that silicon forms this. Silicon loves oxygen. It immediately binds to oxygen and forms sand. sand के साथ कर क्या सकते हो तुम? Sand is not lifelike. It just doesn't behave like carbon's other macromolecular structures. It is just silicon oxide, silicon oxide, silicon oxygen, silicon oxygen, silicon oxygen. It just gets trapped in this thing from which you can't go any further to any other state. But the same thing with carbon. When carbon binds to oxygen, whether in the presence of hydrogen or in presence of other atoms, you form a whole bunch of other very interesting molecules which can both make and break bonds under different sets of conditions readily. You have that dynamicity in the molecule which is completely not there in silicon. So at least under the conditions in which life emerged on Earth, the chance for silicon to result into the chance for silicon to replace carbon as the foundational element, 0 to negative. But like you very correctly pointed out, if in one of those, extraterrestrial body in the presence of hydrocarbon lake conditions, if silicon can behave differently, can it result in another form of life? We need to be open to alternate biochemistries, because we in our own lab have learned that going directly from the prebiotic soup to an RNA world is completely non-trivial. Even though I put it out on that timeline, or I didn't put it out, the other scientists who worked on it put it out. Going directly from the primordial soup of molecules to the fundamental monomers to RNA world is non-trivial. And in our group and a few other groups in the world have shown that the RNA world had to be predated by pre-RNA worlds where the informational molecule fundamentally has the same sets of, it has very similar sort of overarching structure. It needs an information coding molecule, it needs a trifunctional connector like a ribose, and it needs an ionic linker like phosphate. But they didn't have to be exactly what we see today in biochemistry. It could be each individual entity can be replaced and you can still potentially form an informational molecule. And we in our group are trying to understand these alternate biochemistries. And we think they would have very much existed early on under different temperature, pressure and other kinds of environmental conditions. But due to competition during evolution, molecular evolution, they probably lost out and hence you don't see signatures of all of them. However, for example, even in your own cells, you just don't have A, U, G, C, T as the bases. You have a whole bunch of modified bases, which to us is a very strong indicator that your RNA world, pre-RNA world, actually explored more of these heterocycles than what you see in biology today, which was used for information encoding. Today it doesn't, you only see adenine, guanine, thymine, cytosine and uracil in the informational coding business. But maybe early on you had other molecules which were exactly these pyrimidines or purines but had the heteroatoms decorated in different places. As long as you can hydrogen bond with a partner, you can do information transfer. So it could have been a whole bunch of other things. So I think it's very important to be humble and not behave like you figured it all out. And that there could be alternate biochemistries when you're looking for life elsewhere in the universe, and that you have to keep your eyes wide open.
Thank you, ma'am.
Can you talk about us work?
Oh, what I do in IISER? We are fundamentally interested in a whole bunch of processes where we go from the monomers to polymers to forming the protocellular entities. So you can sort of go to our lab and we have a really poorly maintained web page, but it should be enough to give you a sense of what it is that we are interested in. But very quickly, we are interested in how you form the first nucleic acids. Are they what we see in biology today, or do you have other alternate biochemistries? How did you form the first peptides? Because all of them is a very important chicken and egg problem in origins of life conundrum. You need information encoded in a nucleic acid to make a protein. However, you need a whole bunch of proteins to make that nucleic acid. Look at your cell. For DNA to replicate and for using the information in the DNA to make an RNA and then a protein, it's like a mega drama of highly engineered molecules that are doing the job. Very early on, what came first? Right? This is why the RNA world hypothesis is exciting, because it provides a respite. Now you have to worry about how one molecule came about, which would both encode information in some shabby manner, but it did. Viruses today, to date, use it. Your SARS-CoV virus is an RNA virus. It is using information in the form of an RNA molecule, not a DNA molecule. But they also get the reaction. So if you can simplify the problem and figure out the first few steps, then you can think about the more complex things that happen with time. But they're interested in understanding how catalytic molecules came about, the peptides, first peptides. How did they cross-talk with, I didn't touch upon a whole bunch of things, but for example, your hemoglobin molecule can do what it does, which is bind oxygen, only because of the iron present in the porphyrin core, which is present in each subunit. That iron binds to the oxygen. But without the protein, you just take the iron in some hydrate form or the iron coordinated with the porphyrin, try to make it bind oxygen, it doesn't do it very efficiently at all. So this whole thing is very sort of layered in its functioning. How did the earliest of enzymes, hemoglobin at some level is like an enzyme, it catalyzes the binding of oxygen. How did those functions come about in simpler molecules? Just peptides talking to simple scaffolds where your divalent cations are present in either a coordinated manner or not. We try to understand some of those questions too, and we are interested in the membrane. What are the molecules that made the membrane, and how did all of them come together to form the protocell? So we try and think of many different things. Please go to the website, and I'm happy to chat over email.
Last question. Thank you, ma'am, for giving your precious time here and sharing the knowledge with us.
No, it's not my precious time. This is precious what I'm doing right now. But yeah, go ahead.
Ma'am, you said that carbon exhibit allotropic nature and it's tetravalent. So that means it's more reactive compared to radioactive elements or less?
No. So, see, radioactive elements also have reactivities which completely change with the nature of the atom. Some react very slow and their half-life is for several hundreds and thousands of years. Some react much more readily and can decay into, other elements. So that depends upon the atom and certain other properties of how the, electrons and protons and neutrons are sort of doing their drama in that particular atom. I'm not saying that at all. All I'm saying is, carbon when it's bound to say an oxygen, for example, forms a carbonyl group or an aldehyde group or a carboxylic acid group where you have a double bond present. That makes it a much more interesting place for a nucleophile to attack and then facilitate other reactions. So that is where carbon chemistry is very interesting. Carbon chemistry can form a lot of these electrophilic centers where nucleophiles can attack and facilitate other reactions which are very important for biochemistry. Make sense?
Thank you.
Thank you everyone for your time and attention.
Hello, Madam Professor, in fact, before I make an announcement, every rule has got an exception. I want to ask you one question. Actually, I hope all others also will appreciate. During the course of your talk, you told about asteroids and comets. Might have brought the ingredients of life.
Whether they brought it in that kind of quantities that is
assumed
during electricity.
But there is a theory which is proposed by the controversial Professor Fred Hoyle, about panspermia theory. That is, the life itself was seeded on Earth by probably extraterrestrial visitors like comets or asteroids. I just want to know about your opinion on that. It's highly controversial. As you said, unless proof is there, any hypothesis cannot become theory. Just I want to know your opinion.
Is it a reasonable hypothesis? Answer is yes. As loud as I want to be without the mic. It's a reasonable hypothesis at the first principle level. However, my fundamental problem with it is that you're just transferring the problem elsewhere to another entity in the solar system about which you have no clear idea of its geological history. You have no sense of the processes, geological and chemical processes that are playing out there. So it just is intractable beyond a point. Unless we start doing time travel and we can go back in time and figure things out, it's very hard to prove this. But one other consideration which is very important for us to remember is that re-entry process is extremely disintegrating to macroscopic entities. Molecules are a different ballgame. You're talking about smaller angstroms. But if you're talking about microorganisms, you're probably sort of, kind of in that regime, a couple of tens of microns. But I still find it really hard to imagine that they could have survived the early Earth re-entry processes and survived. Already a lot of what molecules got delivered in the form of molecules, exogenous delivery, also could have gotten decimated because of those extreme conditions. The ones which survived in the cores, deep inside, which didn't get, ripped off during the re-entry, that is the one which eventually led to, all of that water and everything else that you see now. So that's my problem. You're just shifting the problem to some other body in the...
Thank you very much. And just before we have formal vote of thanks, I want to make one more announcement. That is, kindly do attend for the next Saturday and Sunday lectures. Already some questions came. See, next Saturday, under this lecture series, that is Dasara lectures of 2025, the next lecture is CO2 Reimagined, Creating Useful Chemicals and Fuels from Carbon Waste, by Professor Sebastian C. Peter of Jawaharlal Nehru Center for Advanced Scientific Research. At the same time, next Saturday. If there is any change in time, we will let you know. And next Sunday, okay, under the same Dasara lecture series, theme being the carbon, the same, Professor Sarit Agasti of IISER, again, he is speaking on designing with carbon, the creative power of organic chemistry. That's what you are asking question. Now, anyway, formal vote of thanks will come. Before that, I would like to request you, after the vote of thanks, you can join for high tea outside. Lot of carbohydrates as well as lipids are there. A bit smattering of proteins is also there. Kindly do have it. Thank you. Formal vote of thanks by Sri Jawaharlal.
Good evening to you all. The present task of delivering the vote of thanks has come to me. In this regard, I express our heartfelt thanks to Professor Sudha Rajamani from Institute of Science Education Research, IISER Pune, for having accepted our invitation and delivered a wonderful talk. Question and answer session was as interesting as the lecture. This signifies the appeal of her narrative to this wide audience. On behalf of GNT and my own behalf, again, thank you very much, madam. As you know, this is the second lecture in the Dasara lecture series 2025. While delivering vote of thanks, it is my duty to thank our director, Dr. B.R. Guruprasad, for taking initiative and providing guidance to arrange these inspiring lectures. Thank you very much, sir. Let me not forget all my JNP colleagues who have contributed their might in making this program successful. And also thank the audience for actively participated with interest for the program. Of course, our thanks will also go to our media friends. I hope the same interest will continue for next two lectures during the next weekend as well as for our other programs. I also thank about 100 people who joined online to listen to Professor Sudha Rajamani. Thank you one and all. Be good, do good. Dhanyavad.